Good morning. Welcome to the Daily Planet inside the Nature Research Center. I am Julie Urban. I'm formerly a scientist up in the genomics lab, but technically I'm faculty at Penn State University. Woohoo! First time I get my affiliation out there. So thanks for visiting us. You are here in a very special time, and for the last 2 weeks, we've had teachers, middle school teachers, working in our labs, working on three teachers each working on one of 4 projects, where this is funded by a National Science Foundation grant for $7 million. And each of these projects is led by a postdoctoral scientist. And for these 3 special weeks, we're in week 3, we have these teachers who work with the scientists to co-create lesson plans and co-create the original research projects, where basically we're engaging students in making real science happen in their classrooms. So these are citizen science projects. This is legit science. It's going to be published in the academic literature, but teachers and students, and I can't call them teachers anymore, there are scientists, because they're doing science for real, and they're going to then engage their students in that science in their classrooms. And so this is a very special project, and today we're going to hear from the first of our teams who works up in my lab, my former lab, genomics, and this is Team Soil. And here you can see the scope of this because we have teachers from Brunswick County all the way to Buncombe County in this team, so 422 miles apart, and a teacher from Wake County. And so let me call them up and they can introduce themselves. And we have Grace, Leah, and Melissa who are going to talk about their project.
So it's loud. I am Leah Goman, and I'm the Buncombe County PEEP. Do you guys want to introduce yourselves right now or when you guys talk? Okay, sounds good. The first thing that I'm going to mention to you is that one of our projects, we're doing a couple of them because we are adventurers, is a maze experiment. So students will be testing a prototype for a brand new tool that will test the growth and nutritional diversity of bacteria. So let me tell you how they're going to do this. Step 1, they have to design a 3-dimensional maze. Step 2, they have to print it out with a 3D printer. Then we get even trickier. They have to make some agar, all right? And then inside of a sterilized petri dish where your 3D maze is already placed, they have to pour in the agar. So after that solidifies, then the students are going to continue to decide what's going to happen in the experiment. So they're going to decide the types of nutrient that they're going to put in the 4 corners of their maze, and then the bacteria will be placed in the center. So they might choose things like human food sources or pollutants or whatever, and so the bacteria will be in the center, and then they're going to monitor how the bacteria grows towards the different type of nutrient. And so they're going to do this for a couple of weeks, and what will hopefully happen is the bacteria that we placed in the center that was delivered by a scientist to the teacher, that bacteria will then grow, hopefully in the direction that through the maze to the food source that the bacteria would prefer. And through all that, the students are going to take daily pictures so that in the end, they can make a final product that has a time-lapse video and then also display their information in a table and a graph to show the speed in which the bacteria grew.
I am Melissa Harris. I am an instructional science coach in Brunswick County Schools. I work with 39 teachers and around 3,000 students through the year. What is amazing about the outcomes of this is that it reaches all 3 grade levels, 6 through 8 in the life sciences. It also meets some of the national next generation science standards. We've incorporated a component of language arts, and we've incorporated a component of math so that we have been able to integrate it throughout the entire curriculum. What's an authentic outcome that we experienced is we had a contaminated stock solution when we started our first section of the mazes, and we realized that at the end of last week. It led to a very authentic discussion as students would have if they had a problem with a scientific inquiry-based project. So we had to come up with alternate things that we would do, and in effect, we had to start the project over, which many of us have experienced in our classrooms, or as students you've experienced in your classroom of having to start again and again and again. That helped us understand what our postdocs and many of the scientists in the real world with the careers that we hope the students will get are having to do with their problem-solving skills. That so many, Julia, our scientist, that is our mentor, has said that she's been many months before having to redo things. So it gives them real life and real world experiences, and it also allows them to see and use critical thinking and their problem-solving skills. So that was our hopes for the outcomes. We experienced them, and we hope that they will transfer to our students.
Hi, my name is Grace Durstein, and I am a language arts teacher. I teach 6th grade in Wake County, and I'm going to share with you a little bit about our second project that we've been working on. So it kind of started out our first week talking a lot about the history of genome sequencing and just how that has become more accessible to the point where now our students are going to get to play a part in that. So we started out with some background, preparing DNA, amplifying it through PCR, and then actually completed gel electrophoresis to take a look at some DNA sequences. And that was really cool because, as you can see the vocabulary behind me, I think what I've been learning is that we're trying to take these words and kind of make them 3D for our students. In other words, we're giving them hands-on experiences through doing these different activities that we've gotten the opportunity to do. So what they'll actually be able to go ahead and do is they're going to receive some DNA sequences of different microbes and plug that into the computer through GenBank, and their result will be different research that has been done on that particular microbe and the function of those different microbes, so they'll learn more about that. But the really cool aspect of that is that they get to report this research back to a scientist who may not have the time to go through all of that. So they're really being given the power to do that research, but the even cooler aspect is that they're taking the material that they find from different scientists around the world and connecting that. So the students have the opportunity, that hands-on experience, to have the power of connecting scientists and helping them to collaborate, which is really cool. And as a language arts teacher, I've actually found kind of a cool connection. What I'd like to do, we talked about this, is taking a poem and connecting the way that DNA sequencing works to a poem and how they can kind of take it apart and put it back together in a new way, and kind of look at the function of a poem, just like we know the different sequences and the different microbes have different functions. So that's kind of our language arts connection and tie-in there. So with that being said, we wanted to just end with kind of our personal experiences throughout this time that we've had in the museum. So for me, it's been really neat coming in as a language arts teacher. I was pretty nervous just kind of coming into this whole science world, but I think it really speaks to the power of citizen science that I have been able to just participate in the labs and learn so much and just be in a totally new environment and just doing things that I never thought I would be able to do. And so I think that what I want to take back to my students is that power, and to show them that science is for anybody, and that they have that power and they can have that control to be able to do really whatever they want to do, and to make a huge impact in the world. And I think our projects speak to that power that they'll be able to have. So that's really been a cool thing for me, and just seeing the tie-ins also of how language arts can connect to science. At first, I think I was kind of nervous about that, but I've just seen so many different connections I think we all have in our different subject areas, and so that's just been really powerful.
Okay, I'm going to ask you guys a question. All right, interactive here. Okay. Do you know what song I woke up singing this morning? No guesses? Okay. It was Disney, not Frozen. Okay? All right? It was Pocahontas. All right? So if you travel back in time and remember when you watched that movie, okay? And do you remember the part where she gets in her canoe and she has her sidekicks? I forget, Miko maybe was one of them. Okay, good. All right, and she starts singing, just around the river bend beyond the shore. You guys know what I'm talking about? You guys remember that? Good. All right. Well, this experience for me personally has been wonderful. It's been quite the adventure, and I think that's why I woke up singing that song this morning, okay? But how many of you on a normal Monday morning, okay, wake up singing and excited to go to work or school? Oh, good. There's a hand back there. Okay. But the rest of you, I'm speaking to you. All right. All right. So sometimes we need a little pick-me-up. We need help getting excited. And we need a fresh perspective. And I really think that this experience with Students Discover and Citizen Science has really given me that rejuvenation factor. Okay, if you can tell. One of the things I realized this morning while I woke up singing, okay, every person, all right, is born to be an adventuring scientist. And you might think, no way, not me, I don't do math, okay? But I believe it started when you were a baby. And you started that hand-to-mouth thing, and you put stuff in your mouth. You put all sorts of crazy things in there. It might have even included lead paint from grandma's walls, okay? I know they've been doing a lot of research on that, okay? But what gets even more exciting is when you, or let's just say in general, a child gets to be about 4 or 5 and they start asking tons of questions constantly. So imagine, this is like a real situation I saw at Walmart, okay? So you're in Walmart, there's this little kid, all right? And as loud as humanly possible, the little kid turns to mom and says, "Why is that woman really fat over there?" And you're like, "What? My kid just said that." All right, quickly, I must explain to my child that this woman is actually pregnant, and that means that she has a baby growing in her tummy, and that's awesome. Okay, but guess what? The questions just keep going. They're like, "What? There's a baby in there?" All right, so when we are little, okay, we have all these questions and this big world in front of us, and we are super excited about it. And through this experience, I've learned a lot of things, all right? I've been reminded of things I learned in high school or in college. I've been introduced to a lot of amazing experiences, but what I have found, like my big thing that I want to take home as a teacher is that we are naturally prone to be scientists. We're inquisitive, we're excitable, but sometimes we stop asking questions either because we don't know what the answer is or we're nervous to ask the question. So I really want to encourage my students going forward to continue asking those questions, investigating those answers, and getting to be real scientists through citizen science, and possibly, if they choose to, pursue a career in science as well.
I have said my entire life that I'm a genetic marvel, not as a superhero, but I am an artist that is blended to a biologist. I grew up the child of 2 educators and went as far north from education that I could. 3 career paths to education. And I'm in a job that I love and hope to have to the end of my career. The reason I chose to do Kenan Students Discover team was because I wanted to be able to translate active science into classrooms and into children's hands. I wrote a reflection in this, and I'm not going to read it, but I equated it as to me going into the experience at Cullowhee and then the experience here, as I was going to have a blank canvas and utilize the scientists, my fellow peers, and all of the people that I met as the hues to color the canvas. I haven't been disappointed. I've had many works of art come out every single day. I'm consistently tabbing in my brain more than just what we're doing. I've already gone back and rewritten curriculum products that we have for our county, and it is beyond any expectation I ever had. Even though I've been in many labs and in many places and met many people, when we did the reflective video, we included the other Students Discover team members, and we included the other postdocs and mentors to our other teams, because we feel like we have taken away from everybody that we have come in contact with. You have been gracious hosts, and we are excited for the year ahead of us. Thank you very much.
Fantastic, thank you. So let's keep you up here, because it's likely that folks in the audience, maybe on different floors, or folks here, might want to ask you some questions. So do we have any questions for We Are Soil? Oh, you'll get the honors.
So you touched on the contamination we had, but what finding did that contamination lead to?
Yes, it was pretty cool, and I think Melissa touched on it for sure. So we had contamination, and we were like, "What?" We were disappointed, like I think most scientists would be when they like work really hard. We're like, "We're gonna make it really sterile. It's gonna be perfect. It's gonna be great." And then we walk in like day 2 or 3, and we're like, "Something's weird here." It grew really fast. But what was really awesome, especially on the one contaminated sample, is that the bacteria that wasn't supposed to be there did actually grow through the maze. And we watched it, like at one point in the maze, there wasn't any food further on, so it just sort of stopped growing. And it really did grow in the direction that we had placed the food in the corners. And so even though that wasn't our plan to have that contamination, and our time-lapse videos didn't work because it grew too fast, it was still a really cool realization that we had, even though it wasn't our original purpose.
Actually, the maze itself, it took the path of least resistance, the one that we had, because they weren't symmetrical, and that led to an academic discussion as well. And we thought the kids would really think that was cool, because we had to come up with hypotheses of, oh, why did it go around that corner? Because they were in all 4 corners. Why did it go around that corner but not go around another? And so it led to some lively discussions, and then we said, okay, we want to do it again. We didn't want to quit, which I think kids don't want to quit. They want answers. In fact, a lot of us, we said before, when we did our time before here, we said, you could Google that, or, what do you think? And so I think it gave us an authentic student experience that we might have, because we have already got plans for implementation of the lesson in our classrooms. So, yes.
So I think all of us who are a part of this project are so excited about the sort of way you guys are talking about translating it back to the classroom, and how it's enhanced your career and continue to grow you all as professionals. So that was so exciting to hear that. I did want to ask us a science-y question. What is your favorite, and maybe all 3 of you or just a couple of you, what's the most interesting piece of science you learned since you've been here that you just thought was so cool?
I am the oldest of the group, and I actually did genetic sequencing as an undergrad at Carolina on the first genetic sequencing machine, which was about how many times? 10 times bigger than the ones we have now, and much more expensive. And we literally we were using whiteboards to write on the sequencing that we got with the tobacco virus that we were studying. To see the technology that has gone from there to the technology, the database, the GenMap, the magnitude of research. Personally, I lost my mother to metastatic breast cancer, and it makes me, I get chills. It thrills me the way the technology has just, in genetics, has just come from, and that's not been that long ago. I'm not that old. but the amount of research and genetics research, it makes me excited. It makes me excited for many reasons. Environmentally, which is a lot to do with the soil and environmental implications, medical implications, all sorts of things. It truly gets me excited from my biology scientist standpoint.
I'm probably stealing Leah's, but the gel electrophoresis was just really cool for me because I guess the first time we did it, our results didn't show what we wanted it to show, so we had to do it over again. But to be successful in that lab was really powerful for me because I wasn't always very successful in labs in school, and I feel like I didn't have a lot of like teacher support, I guess. So that was just really cool experience for me to go through and actually make the whole thing and watch and see the results of it.
So I'm going to answer your question, but it might not totally sound like it. What I really enjoyed the most in the lab would be, well, I was going to say gel electrophoresis, so I had to think quick, okay? But I really loved working on a team. I feel like a lot of scientists, they might sort of individually work on a project, but I know like Dr. Julia Stevens, she has, interns that are both, in college and high school, and she works with a lot of people on a lot of projects. So she has like this big team of people. There might only be a couple of people around at a time. But I would say, especially our first week working together as a team, we were really on it. We were considerate of each other. We worked well with each other. We would do a gel electrophoresis, but we can't all have our Jello molds all at once. So like we would trade off. Like I would do something, then I pass it on to Grace, and then Melissa, and then we go around. We just shared everything. We shared all those experiences, and I really enjoyed one day when we were plating bacteria. And when you do that, you don't actually see any of the bacteria right away. It takes a little bit. But here we are, one day we're diluting some samples, and then the next day we made plates with agar in them, and then here we're dribbling the samples down, we're labeling the plates, we pass it on to the next person, we twirl it around to spread the sample, and boom, we're done. And I felt like as a team, we really worked well together. I mean, just coming together, we did not know each other at all, and just the opportunity to work with brand new people, get brand new ideas, and at the same time do science, I think was just amazing. Any other questions?
Let's hear it one more time for We Are Soil. You guys did great.